boleh ah? Ha. Okay. The secretary not here.
Ada doktor Ko. Azwin ada dekat sini.
Boleh siapa-siapa tolong call Dr. Venu.
Okey doktor.
Doktor, transfer dulu saya sebagai host.
Siapa host?
Sekarang doktor Ko host.
Saya host.
Jadikan sebagai mixed mode. Inform ni sebagai host dulu. Nanti saya transfer doktor sebagai co-host. Doktor klik kita baca yang apa dot. Then tukar kepada saya sebagai host.
Siapa? Sekejap Puan Avin macam mana? Nak tukar host ah?
Doktor nak tukar. Doktor nampak dah ada yang 3 dot tu. Tepi belah kanan sebelah nama doktor ada yang dot-dot tu.
Ya.
Okey, doktor klik situ.
Host.
Mixed mode.
Doktor klik dekat mix mode. Doktor nampak ada mix mode kan? Sekarang mix mode co-host. So mix mode kena jadi host. Doktor pergi mix mode. Doktor nampak ada dot. Dan doktor klik.
Mix mode klik mix mode.
Okey.
Mix mode host.
Okey, yes betul.
Boleh sekarang?
Betul dah doktor, betul.
Thank you.
Thank you doktor.
Yan, you can start.
Can you hear me?
Dah start.
Yes, very clear.
Okay. Can everyone see the slides in the presentation mode?
Yes, I'm okay.
All right. So here I begin already. So good morning, everyone. Today I'll be presenting to you on the title of Expression and Purification of TCR-like Antibodies Against Latent TB in Bacterial System. And this final presentation is in partial fulfillment of the TFM 531 subject under Molecular Medicine Mixed Mode course. I'm Yi-En and under supervision by both Dr. Tai and Dr. Kumita. LTBI is defined as the persistent immune response towards MTB antigens without evidence of TB symptoms, and it is important to detect LTBI as 1/4 of the global populations were estimated to be infected with this disease, and 5 to 10% of these individuals will eventually develop TB disease during their lifetime. And hence, this makes the screening for TB to rule out TB disease and the testing for LTBI important as to achieve the global targets of an TB strategy. Currently, we do have a widely used diagnostic tool for detecting LTBI, which includes TSC and IGRA. However, both of these tests come with certain limitations as they do not provide a direct measurement against NTB antigens and also do not differentiate between ATB and LTBI. While these limitations continue to impede the accurate diagnostics of LTBI individuals, and it poses a significant challenge to the global TB eradication plan. Hence, a new diagnostic tool that could overcome these limitations are important and is a game changer. And one of these entities could be TCR-like antibodies, which possess ability from both cellular and humoral arm of our immune system. Shown in studies, it is able to both recognize peptide MHC molecules and elicit the effector functions of antibodies. Previous study by my senior has already developed and discovered this TCR-like antibody generated through phage display technology, namely A2, A11, and A284. The functionality of these antibodies was also tested against the pMHC molecules, where MHC covers 70% of the Malaysian population. The peptide molecules originated from a 16 kilodalton heat shock protein, which was primarily produced during NTB dormancy. Apart from that, it accounts for 1/4 of the total protein that was expressed during the dormancy period. Studies suggested that detecting this heat shock protein has the potential to differentiate individuals with LTBI from ATB, suggesting their diagnostic applicability. As promising as it sounds, TCR-like antibodies are able to overcome the limitations, whereby it provides a direct measurement of MTB antigens and also differentiate between ATB and LTBI. These studies, I am trying to bridge the gap between the TCR-like antibodies generation and discovery to the point of care diagnostic application in the near future. Hence, we are trying to express this TCR-like antibody in a fast and cheap way in preparation for large-scale production. While there are so many different expression systems in the market with the advantages and disadvantages as shown in these slides, however, why are we doing a bacterial expression system? The answer is easy, as many studies have shown that bacterial expression system top in cost efficiency, ease of use, speed, and protein production capacity as compared to other expression systems. And this aligns with the goal of our study, which is to produce antibodies in a fast and cheap way. Apart from that, single-domain antibody was shown to have a very small size of approximately 12 to 15 kilodalton and a lack of a Fc chain, which can simplify post-translational modification requirements that are lacking in bacterial system. And this makes bacterial expression system suitable for the expression of single-domain antibody in our case. This leads to the objective of our study, which is to optimize TCR-like antibody expression in E. coli expression system with the specific objective as shown in these slides. So first of all, I have to clone TCR-like single-domain antibody into a suitable bacterial expression vector, followed by the optimization of the bacterial expression and purification condition. And last but not least, the functionality of the antibodies that are produced will be tested. The overview of our methodologies are as shown here. The cloning will be done, and optimization, the expression of the TCR-like antibodies will be optimized under different induction conditions, including the temperature, incubation time, and IPTG concentration. And the purification will be optimized through immobilized metal affinity chromatography. And last but not least, an ELISA was performed to confirm the functionality test of the TCR-like antibody that was produced against their kinetic antigen. The overview of a molecular cloning are as shown in these slides. And briefly, we have performed a gradient PCR prior to performing a DNA amplification with Q5 high fidelity DNA polymerase to determine the annealing temperatures of the primer. And here we can see there is a consistent band across all temperature that was tested at approximate size of 1 to 1,3 base pairs. And here we chose 50 degrees Celsius as the annealing temperature for the subsequent DNA amplification with Q5 high fidelity DNA polymerase. And here we can observe a single band that was amplified at 1 to 1,3 base pair and 1 to 9,8 base pair of both A24 and A11 single domain antibody gene fragment. And with this PCR product, we perform a double restriction enzyme digestion with NOT1 and NCO1 enzymes. And here we can observe a target band at approximate size of 297 base pairs, which correspond to the A24 single domain antibody fragment. And also, we have also performed the similar test on A11 single domain antibody, which we restricted with the same enzymes. And the target band at approximately 389 base pair was observed, which corresponds to the A11 single domain antibody gene fragment. With this, The DNA was extracted from this gel and ligated into a PEP51B vector, and followed by that, a transformation was performed into a bacterial host cells. Subsequently, the positive clones were selected and sent for a DNA sequencing, which both A24 and A11 single-domain antibody positive clones showed 100% identity against their respective original sequence. With this, we proceed for optimizing the protein expression and purification condition. This slide shows the SAS page analysis results for expression of A11 and A24 single-domain antibody in both T7 shuffle and BL21DE3 bacterial host cells with the expression condition as shown on the right. Here, we are unable to express A11 single-domain antibody where there is an absence of target band at approximate size of 16.4 kilodalton in the post-induction as compared to this pre-induction control. However, we are able to successfully express A24 single-domain antibody in both these host cells at this expression condition, where there is a target protein size at approximately 13.7 kilodalton in the post-induction as compared to their pre-induction control. In order to express A11 single-domain antibody, we have tried other incubation conditions for both host strains. However, none of these expression conditions are able to express A11 single-domain antibodies. And due to time limitations, we set aside A11 single-domain antibody expression and proceed to focus on A24 single-domain antibody protein expression. This slide shows the SDS page results for expression of A24 single-domain antibody at varying IPTG concentration, temperature, and incubation time in T7 shuffle cells. The first vial of each gel was loaded with uninduced bacterial pellets where no IPTG was added as a pre-induction control. The subsequent vial was added with bacterial pellets that were induced with different IPTG concentration ranging from 0.05 to 1 millimolar. As shown here, A24 single-domain antibodies was expressed best at 25 degrees Celsius upon overnight incubation and 37 degrees Celsius upon a 6 hours incubation duration. Other incubation duration and temperatures only show the minimal protein band at the target size, comparing the post-induction to their pre-induction control. With the protein being expressed, we have also tried to confirm if the his fusion tag was expressed together with a protein. And here we perform a Western blot analysis for the cell pellet for expressing A24 single-domain antibody, which was detected by the HRP conjugated his probe. And on the start nitrocellulose membrane, we can observe a single darkened band at approximate size of 10 to 15 kilodalton, which corresponds to the target expressed protein in the SDS page results. And here we confirm that the R protein was expressed alongside with his fusion tag, which is important for the purification step. And next, we also perform a protein solubility testing with a dot blot analysis. Briefly, the soluble and inclusion body fractions of a cell expressing A24 single-domain antibodies at different temperature and incubation duration was extracted and tested. While we can see that different temperature and incubation time, the protein was consistently produced in the inclusion body fractions, even at a very low IPTG concentration of 0.05 millimolar. And this suggested that the subsequent purification step has to be done in the denaturing condition. So a purification was performed on NiNTA resin with 8 molar urea as a solubilizing agent. And here on the SDS page results, we can see a target band at approximate size of 13.7 kilodaltons in the inclusion body fraction, which the results were validated by the dot blot analysis result as a darkened spot on the nitrocellulose membrane. However, in this purification round, we can see a loss of target protein, which was indicated by 13.7 kilodalton band observed on the SDS page results in flow through wash 1 and wash 2 fractions, which were also validated with the dot blot results as a darkened spot on the nitrocellulose membrane. Due to the loss of target protein in these fractions, the concentration was so low and was determined at 0.00054 mg per ml. And in order to increase the binding efficiency of this his tag target protein onto the NiNTA resin, we have also tested different purification buffer at different pH and NaCl concentration. However, in all these tested conditions, the protein yield is consistently less than 1 mg per ml. Per liter. Hence, we go one step back to assess another denaturing agent, which is guanylium hydrochloride, which are commonly used in the research settings. Here, we run a small-scale testing to compare between urea and guanylium hydrochloride as a denaturing agent for my target protein. PBS serves as a negative control, and after 2 hours of solubilization, 8 molar urea shows a decrease in cell pellet size as compared to the negative control. However, for 6 mole of guanidinium hydrochloride, it shows an excellent solubilizing ability as there is an almost complete dissolution of the cell pellet as compared to the 8 mole urea and PBS negative control. With this, we perform a purification with 6 molar guanidinium hydrochloride, and we finally able to retain most of the target protein within the column during the purification step. And here it is indicated by the non-observable band or the very thin band in the flow through wash 1 and wash 2 fractions. With this, we concentrate the protein to obtain a concentration to 2.21 mg per ml with a yield of 11.05 mg per liter. In the concentrated purified fraction, we are able to obtain the target band size at approximately 13.7 kilodalton. However, this purification was suboptimal as there is a presence of co-purified contaminants at the size of 40 kilodalton and above. And hence, we increase the washing stringency of the buffer by lowering the pH and increase the imidazole concentrations as indicated with additional wash and in wash 3 and wash 7. However, we can see on the dot plot analysis results that there is presence of a target protein loss at wash 3 and wash 4 fractions indicated by the additional wash at wash 3 that we have added in this purification round. The concentration of this purification was drastically dropped to a 1.311 mg per ml and a yield of a 0.550 mg per liter. Although we are still able to obtain our target protein at this stage, however, the co-purified contaminants persistently exist in the purified fractions. Hence, there is a need for the further optimization of the washing conditions for purification. This table suggests tabulate all the protein purification condition and the protein yield that was obtained. Notable that when we are using guanidinium hydrochloride as a solubilizing agent, we obtain an almost 5 to 10-fold increase in protein yield as compared to the urea buffer that we are using for protein purification. And last but not least, we perform a functionality testing for A24 single domain antibodies against their kinetic antigen and showed the significant binding of these antibodies against their target antigen as compared to the non-target antigens. As for discussion, there are 3 main parts to discuss for these studies, which includes molecular cloning, protein expression, and protein purification. For molecular cloning, we have successfully cloned the A11 and A24 single domain antibody gene into a PET51B vector, as validated by the sequencing results. Although we are able to clone this A11 single domain antibody gene into a PET51B vector, however, we are unable to express the A11 single domain antiprotein in both T7 and BL21 cells. And this might be due to the presence of rare codons in human proteins that are infrequently used by bacteria such as arginine, isoleucine, leucine, and proline. Apart from that, the frequency of codons in the DNA sequence can be positively correlated with corresponding tRNA species, and the tRNA concentration can determine the number of amino acids available for protein translation, and which in turn it will affect the efficiency of the protein synthesis. This can explain the very low expression or the complete failure to express the A11 single domain antibody in both bacterial host cells in our study. On the other hand, we are able to express A24 single domain antibodies. However, although studies have suggested that reduction of IPTG concentration and the lowering temperature can be beneficial for protein folding, for extracting a soluble protein. However, we are unable to produce A24 single domain antibodies was consistently produced in the inclusion body, even when we are trying to express it in very low IPTG concentration of 0.05 millimolar. And this might be due to the lack of post-translational machinery in the E. coli system and combined with a high reductive environment in the cytoplasm, which can inhibit native disulfide bond formation. Apart from that, the use of a T7 promoter, which is a strong promoter, and it can tilt off the balance between the expression rate and the protein folding rate and leads to the formation of protein aggregates in such case. And last but not least, for protein purification, we are observing a very poor target protein binding towards the NI-MTA resin, which might be due to failure of 8 molar urea in completely disrupting the inclusion body aggregates. While inclusion bodies are defined as the nuclear cytoplasmic periplasmic aggregates of biomolecules of mostly misfolded proteins, many different protocols was developed for the extraction of different inclusion bodies. In this study, we are using urea and guanidinium hydrochloride, which are commonly used protein denaturing. And studies have shown urea and guanidinium hydrochloride works in different mechanisms in interacting with differently with the protein, hence denaturing the protein. However, it was also shown in studies no single type of solubilizing formulation could disrupt all the interaction between within the inclusion body aggregates. And this could explain why there is a suboptimal solubilizing ability of the 8 molar urea. And this suboptimal solubilizing ability could affect the inclusion body, where the histidine tail can be buried within the inclusion body, leading to the inefficient binding of his target protein onto the NI-MTA resin. While studies have shown that washing with large amount of low pH or higher imidazole concentration buffer can decrease the co-purified contaminants in the purification. However, the washing step that we are used in the wash 3 was a pH 5.3. Studies have shown that a pH 5.97 is enough to protonate 50% of the histidine molecules, leading to decrease in interaction between the histidine tag to the NI-MTA resin. Hence, this explains why there is an illusion of our target protein in wash 3 fractions. And hence, we suggest a further optimization to be done to obtain a more purified target protein product. As a future recommendation, we always strongly recommend to produce soluble proteins, as it will always recover a better yield as compared to an insoluble protein. For this, we are recommending to re-optimize the molecular cloning by considering alternative vector and host cells. So here, we can consider different fusion tag that could enhance protein solubility, alternative weaker promoters, or alternative host cells to be used. Apart from that, we can also reconsider the use of other purification resin, such as talon resin, which can also bind to polyhistidine tag and shown in many studies to reduce co-purification of contaminants. And apart from that, we can also add one step of purification where size exclusion chromatography can be used to remove larger co-purified protein. And as a conclusion, although we are unable to produce A11 single domain antibody gene, A11 single domain antibody protein in bacterial host cells, but we are able to successfully clone and produce a functional A24 single domain antibody in the bacterial expression vector. And however, a purification shall be performed, the optimization of a purification shall be performed for the washing condition to obtain a more purified product. And this is the references. And thank you.
Thank you, Ian. Comments from evaluators? If I follow the list here, Dr. Vanu is the first evaluator. So can Dr. Liao comment after Dr. Vanu? Dr. Vanu?
Yes, okay, sure.
Vanu. Yes, Vanu. Or maybe Dr. Liao, you can go first.
Okay, so Ian, okay, well done for your work. I can see that you have put a lot of effort, and one thing is even we face some not all the project can run so smoothly, and at least you able to find some solution. All right. So compared to your last presentation, I've seen.
Can you hear me? Sorry, Dr. Liao. Can you hear me?
Yeah. Yes, please go ahead, Dr. Venu.
Yeah. Sorry. I think my mic was muted and I was talking. Go ahead, Dr. Liao. Go ahead. Then after you, I'll do it. Okay.
Okay. Thank you. All right. So compared to your last presentation, I see there's a big improvement. Okay, especially your works has been more progress, and your slide has been more focused on the expression. right now. So can you please go to the, I don't want to say, ask for any technical question first. Okay, let's us to like do some, share some study. Okay, why do you want to choose this TCR antibody as your main target in your research? Here,
Come again, your question. Why do we
why,
choose
yeah,
this?
why do you choose TCR antibodies?
Why do I
And
choose?
because you mentioned about the T cell and also the antibodies, and so maybe why do you choose TCR like antibodies?
Why do I choose?
Yeah.
This question is because it was able to recognize PMHC molecules while elicit effector function of antibodies. So this is why we choose TCR-like antibodies.
Okay. So
Okay.
maybe because this is a like sort of the single domain antibody, right?
Yeah.
And what is the big difference of this antibody compared to T cell and B cells antibodies?
Single domain antibodies, of course, they do not have the Fc region actually, and then hence they are different from the antibodies from B cells. And of course, TCR-like antibodies are able to detect intracellular antigens as compared to normal antibodies. It only detects extracellular antigens. That's why this is different from how TCR-like antibodies is different from B cell and T cells.
Okay.
Mm.
And now we go to the discussion about your topic. Okay, expression. Your focus is on the expression, right?
Mm-hmm.
Okay. Why do you choose PET51B? Am I right? Am I correct?
Correct.
As your expression vector in this study, and there are plenty, like for example, 28, 32, and 15. Yeah,
Okay.
apart from your delivery in your lab, and there must be a reason why you want to choose this vector.
Because this vector comes with a his tag, and we want to try to express this protein at with a his tag. Then secondly is because, yeah, what you said, because this expression vector is readily available in the lab. That's why, with consideration that we want his tag and it is available, that's why I chose this PET51B vector. Yeah.
But I think most of the PET vector, they contain the his tag, right?
Yeah, but it's not really readily available in the, and then also this PET51B vector has 10 times his tag as compared to other vector might have 6 times his tags only, because we want to try to increase the binding affinity towards an anti-resin and some sort. Yeah.
Okay. And because you mentioned that your most of your protein are presented in the inclusion body, isn't it?
Sorry, come again, your questions?
Most of your
Uh-huh.
proteins,
Uh-huh.
your recombinant proteins,
Mm-hmm.
they are expressed in the form of inclusion body. They are not soluble, right?
Yeah.
Okay. So I'm happy that you're able to use this top plot to study about these inclusion bodies.
Mm-hmm.
Can you like try to explain why the protein can be exist in the inclusive body?
Oh, this I go to the discussion slides. Yeah, low here. Okay, shown in many studies actually, if we can actually switch around with the expression conditions, such as reducing the IPTG concentration or lowering the temperature, it can help to reduce the inclusion body expression. Then inclusion body expression is due to the misfolding of protein within the bacterial host cells, and it aggregates to become protein aggregates, which was known as inclusion body. And the primary factors that can contribute to the formation of inclusion body in our study is due to the lack of post-translational machinery, and are as shown on these slides, which could be one of these reasons or the combination of these reasons. Yeah, so this is why our protein are expressed in the inclusion body. Yeah.
All right. Thank you, Ian. And the further question is like, there are few layers of the protein can be turned into the inclusion body, for example, cytoplasmic and other plasmic. So
Mhm.
what do you think where the location of your protein can be found in the E. coli before it turn to, like for example, now you express it in the, inside the E. coli, right?
Mhm.
Okay, unlike you use the mammalian expression system where you can get the solid protein easily because it's got the Golgi apparatus and all this kind of
Mhm.
pilipala things. Okay,
Mhm.
and in which layer you suggest that most of these inclusion body can be found in the E. coli?
Okay. Initially, theoretically, single domain antibody will be produced in the periplasmic layer. However, in our study, we found that if I do periplasmic extraction, it will not get any protein that was detected. So we try to sonicate it and obtain extract everything from inside of the cytoplasm, and we get something. So here we can see that the inclusion body is in the cytoplasm.
So
It's not
have
in
you
the periplasm.
so have you tried out in the polyplasm layer?
Yes, there's nothing there. I tried already.
So,
Then because I don't want to waste more time to
yes,
test on that, so I just proceed for sonication.
yes. Okay, maybe so this is like what you say, because since that you're working on the polyplasmic already, how
Mhm.
did you observe your protein in the polyplasmic layer?
Observe. Usually, last time what I did is after I do a periplasmic extraction, I'll perform a SDS page SDS page analysis to see if the protein was extracted in that fraction, and then usually I will also perform a dot plot to confirm the SDS page result. But SDS also don't have already, so
Okay.
there's no point for me to confirm with dot plot. Yeah, the SDS page result, so nothing.
Okay. Now can you move to your slide in the protein concentration?
On protein, this one.
Concentration is concentration. I think that you have one slide show that you purified the protein in 20 over milligram and or whatnot.
This actually is all tabulated here. Protein concentration for one of the protein
Yes.
purification round is this.
Okay, because that from this view that it seems like your protein are able to purify well and concentrated into a certain
Concentration, yeah.
volume, right? And
Mhm.
so from your perspective, okay, since
Mhm.
you are able to concentrate your protein really well, and what is the main problem you face in your project? Because I can see that in the discussion you try to find a lot of the solution, say you want to overcome your very low yield proteins and everything, blah blah blah. And what is the main what is the main outcome you can what is the main problem you face during this project?
Main problem that I face because of the low concentration actually is when I use, because our lab don't have guanidinium hydrochloride initially, and then I keep trying urea and then I keep trying urea and it's so disappointing. All the protein concentration are very, very, very low until even I perform SDS page results for the concentrated protein, I cannot see any band. I think it's because the protein concentration too low. Until only I buy guanidinium hydrochloride, then I can see a protein concentration. Yeah, that was devastating. Emotionally, it was devastating when I tried to purify with urea. Yeah, this is one of the most challenging part of the protein purification step.
Okay, so in this guanidinium hydrochloride, right, do you use any control to compare your recombinant protein with it? Like for example, before adding in the guanidinium hydrochloride and after adding in, have you seen any big difference?
Before adding in the guanidinium hydrochloride and after adding in.
Yeah, it's a native recombinant compared to the have you done this study?
I got, but I don't have the slides ready here. But
Yeah.
soluble region memang have nothing because you see here, you see in all the SDS page results that I obtained, you can actually see there's actually no target protein in the native stage, the
Yes.
soluble region. So I also tried like just to try out if it really purified nothing out. So I actually tried once to purify the soluble region and found that there is nothing. Everything is clear inside the soluble region. So that's why I proceed for denaturing no matter what. Yeah.
Yeah. Okay, in case like in future, this protein can be, is potentially to be turned into the translational form. Do you find that the denaturing method is the best method can be used for the next step of your study?
No. Okay.
And why?
Like what I recommend in the future recommendation, I will always strongly recommend the production of this protein in a soluble region, because I think with the current yield that I'm getting is not I didn't push it enough to get the most out of my protein, the most out of my bacteria that I express, because due to that the fact that I produce this protein in the inclusion body, the denaturing and then folding and each and every step can lead to a protein loss. And hence, we can only obtain, I don't know what is the real yield of this protein, but with this verification, we only get a very low yield. And so in studies that soluble protein can always get a more yield as compared to the denaturing condition. That's why I will always strongly recommend to produce this protein in the soluble region.
Okay, good. And so my last question to Ethan, Ian, it's just for the understanding, not any questions. Okay. As you just now one slide that you mentioned that this protein is not relevant, hardly to be expressed by E. coli because of the certain codon, right? Okay, how do you relate it to your question just now about the stability? How do you yeah.
Okay, actually, usually people when they do in bacterial expression system, they'll try to codon optimize the thing. But in my case, I just cut from the mammalian expression vector and straight away paste it into the bacterial expression vector without optimizing the thing. That's why I think this can also cause the inefficiency for translation, and in some case it can also cause the truncation of the protein when we are expressing it, or the low expression level of this protein, that which we can see in our study. The A11 is the example.
Okay, good. So actually, I still have a few questions. I think it should be okay. I give it to Dr. Venu. But thank you so much. It's a very good
Go
work.
ahead,
Ian.
Dr. Heng. Go ahead.
I'm done already. Thank you so much. And I find that Ian is a little enough. Okay. So congratulations to Ian. So thank you, Dr. Koo. And please go ahead, Dr. Venu.
Yeah, Ian, I think mainly most question was cleared by Dr. Heng. Here that what I wanted to ask you is that as what that you have been saying that you've been keep on using urea, and so do you know? Okay, can you just explain that? What would be the main reason that by using urea you don't get it correctly?
Okay. It was because urea does not solubilize the protein aggregates well. As you can see, people actually study protein. How do I put it? People usually put guanidium hydrochloride or urea as a denaturing agent, and here they always study that how small or how efficient is the urea or guanidium hydrochloride being able to solubilize the cell pellet as compared to its negative control. So this is why I did this experiment to show that urea actually is not as good as the guanidium hydrochloride in solubilizing my protein. And with that, because you can see the evidence here that urea is not as good as guanidium hydrochloride. Hence, they can cause the histidine tag to be hide inside the inclusion body because it is not solubilized well. Hence, this can lead to the hidden his tag within the inclusion body can lead to the inability for this his tag to interact with the Ni-antiresin. That's why 8 mole of urea is not so suitable for the solubilizing my protein in this case.
Okay, okay. No worries. Can you go to slide 46? 46 or...
Okay.
Okay, yeah. So now solubilization refolding a variety of inclusion bodies using novel formulation. So with these things that you have done with this, and now that you have got the quite a soluble and correctly folded protein, am I correct?
Mm-hmm.
So how sure you are that protein that you get is correctly folded? And have you done checkings that
Yeah.
protein is correctly folded?
Yeah, we did actually functionally test for A24 single domain antibody against the kinase antigen, and we found that it can bind significantly onto the antigen as compared to a non-carrier antigen.
So this antigen is the antigen that you have it in vitro, correct? It's not on the cells or...
Yeah, it's not on the cells, unfortunately. It's in vitro antigen that we produce. Mm.
So your target, you get 0.25, your non-target. So with around 0.25, can you consider it that the binding is strong?
Okay, the results here might be due to the different, how can I say? I can say that it is significant because when you do a t-test, it really shows a negative p-value of less than 0.05. However, because when you see here, I use this as the thing for the testing, and this concentration might include all the concentration of the proteins as shown here. So when I am adding this one, it's like I add, but I don't know the real concentration of this target protein that I added into the ELISA plate. So it might be one of the reasons it leads to a very low OD value that we are seeing here.
Okay. So I think the rest been covered by Dr. Heng. So thank you. Good luck to you on your thesis submission and viva later.
Thank you, doctor.
Okay, good luck as well, huh? Ian.
Okay, good luck to Ian. So can I pass the sessions to Dr. Kumita?
Can, thank you, Dr. Koo.
Comel Dr. Kumita cakap.
I stopped sharing.
All right. Okay, we are done. Thank you all. Bye-bye.
Hi, thank you, everyone.
Thank you, everyone. Thank you, doctor.
Okay.
Dr. Shahida.
I think, yeah, we're still waiting for Dr. Shahida.
Okay, I will message Dr. Shahida.
You already messaged?
Yeah.
Okay. Ziyu, maybe did you want to start sharing your slides?
Hello, Dr. Kumita.
Yes. Hi.
Yeah, I just want to
Hi.
test my mic. Okay, thank you.
Okay, all right. Did you want to start sharing your slides? We'll wait for Dr. Shahida, but if you want to project your slides, you can do that.
I can start my slide.
We are still waiting for one of your examiners, but you can share your slides first for now. Maybe
Dia
just
dia pandai dah. Itu dia tak tahu dia.
Wait me a minute. Wait me a little.
So once you have your slides open, you can click on the button share and the tab below.
Mm.
Okay, we can see your screen. Okay, great. We can see this. We're still waiting for one more of your examiner, so we will get started when she is here. Hopefully that's okay with everyone.
Dr. Shahida on the way.
On the way. Okay, thank you so much.
So I start now.
Not yet. We are still okay. Wait, I think Dr. Shahid. All right, good morning, everyone. So we're here for the final progress presentation of CU student of Associate Professor Dr. Koo. The examiners for today's session is Dr. Chu and Dr. Shahida. As you get 20 minutes to present your work, followed by questions from your examiners. You can start whenever you're ready.
Oh, okay.
Yeah, you can start.
Thanks for Doctor and Professors very much for attending my speech. Also, you and my supervisor is Dr. Wu. The title of my final project is the growth inhibitory effect of quercetin on cancer cell lines. Cancer has become an increasingly serious issue in today's world, which is why this research is important. The World Healthy Organization says there will be an estimated 20 million new cases of cancer and 9.7 million deaths in 2024 globally. About 1 in 5 people will develop cancer in their lifetime. About 1 in 9 men and 1 in 12 women will die from cancer. Cancer has become the second leading cause of death worldwide, especially in economically underdeveloped areas. Those two figures show the incidence and mortality of cancer worldwide and their relationship with cancer. Now we need to know quercetin. Quercetin is a plant flavonoid belonging to flavonoid group of polyphenol and has strong anti-cancer properties. Quercetin can effectively inhibit the growth and the proliferation of virus cancers. Quercetin can also reduce cell viability, autophagy, and autophagy or promote apoptosis by regulating signaling pathways. Next, we need to have some understanding with the NCI-60 and GEO. NCI-60 is an in vitro drug discovery platform representing 9 tumor types, which is used to continuously screen potential adjuvant drugs and study drug responses. The GEO stores the genetic data of target cell lines we need. About my problem statements, I found a large number of epidemiological studies highlighting the potential of quercetin to reduce cancer cell viability, growth, and proliferation. However, only a few cancers were focused on. More human cancer cell lines that might respond positively to cancer treatment have yet to be elucidated. As such, our preliminary study aims to investigate the effect of quercetin on 60 different human tumor cell lines using the information in the NCI-60 database to identify and characterize different cell growth inhibitory effects upon quercetin treatment. The effect of quercetin in the NCI-60 cell line panel is well documented but not well studied yet. It's my third question and hypothesis. Can the statistic panel consider cell lines provide enough information for selection of the best cell line upon casting treatment? What is the gene expression of select cancer cell lines upon casting treatment? What are the levels of the growth inhibitory effect of the casting-rich subtraction of rhodanium pure ethanolic crude extract on the selected cancer cell line compared to pure casting? What are the levels of Ki67, PCNA, and PGP gene expression in extract on the selected cell lines compared to pure casting? This study hypothesized a new approach to identify a cell line from its panel or cancer cell line that exhibited the best effects upon casting treatment. The gene expression profiles associated with the growth inhibitory effects of casting on the cell lines could then be analyzed based on the GO repository. Compared with pure casting, the growth inhibitory effects and some gene expression of extract can also provide a fresh perspective on cancer research. My main objective is to comprehensively identify the growth inhibitory effects and its related gene expression of casting on cancer cell lines. It is split up to three specific objectives to identify the cancer cell line that shows the best growth inhibitory effects upon casting treatment in the CI60 cancer cell line panel. To analyze the gene expression associated with the growth inhibitory effects of select cancer cell line in the GO repository, to investigate the growth inhibitory effect and a few gene expression upon the treatment with casting-rich subfraction of rhodanium pure ethanolic crude extract compared to the pure casting in the selected cell line by cell-based proliferation assay. This is my methodology. To approach all the objectives mentioned just now, I first brought the NCI60 and the GEO. The NCI60 database was first accessed through the NCI DTP to select the cell line showing effectiveness upon casting treatment. High throughput functional genomic data of selected cell lines was studied through the GEO database of NCVI to annotate gene identify dataset title links to supporting information and graphs describing the expression level and the rank of gene in each sample in the dataset were included and studied. After that, the selected cell lines will be cultured and treated. The cell will be treated with casting-rich subtraction of rhodanium pure ethanolic crude extract. And for comparison, pure casting will also be used to treat cells. After treatment, the treated cell will be subjected to anti-proliferation assay like TT or LDT assay. The cell morphology after the treatment will be also be monitored under a microscope. The treated cell will also undergo total RNA extraction and cDNA synthesis. The cDNA will be used to detect the identified gene from GEO by semi-quantitative PCR. Finally, we use the GradPad software to make all plots and statistical calculations. the data will be presented as the mean plus or minus standard deviation of at least two independent experiments. P-value less than the 0.05 will be considered as a statistically significant difference. This is my CS60 results. This figure shows the dose response curves of consisting against the panel when CS60 cell lines. Most of the cell line display growth inhibitory effect upon consisting treatment. I also managed to select a few cell lines that exhibit the promising growth inhibition upon consisting treatment. Those are CCRF-CEM with the cell growth level shown at only 55%, K562 39.1%. CIH 522 46.4%, SF 539 44.5%, U 251 44 MDAMB 435 46.4%, A498 48.4%, and HS 5780 57.2%. Based on GI values among the selected cell lines, only CCR-CEM is sensitive, but K562, CIH 522 and A498 are resistant to the consisting treatment. SF 539 and U 251 showed no effect on treatment, and no information was found on MDAMB 435 and HS 578T cell lines. The KI 562, NCIH 522, A498 and CCRF-CEM cell lines will be used for my next studies. And this is my GU results. Based on this set submitted to the GEO database for a project entitled Costing Effect on Gene Expression in HepG2. The data set will divided into groups, into two groups. When treated with costing and the other serving as the control group, treatments were applied for 2, 4, 12, and 24 hours. This distribution of the two selected genes in this set represented by the violin plot indicates slightly different data in the consisting group compared to the control. The same phenomenon of differential expressed genes was observed in the selected genes in the heat map. The graph widely presents the difference in gene expression, specifically the observance level of three selected genes, Ki60, PCNA, and PGP under those conditions. Log fault change was used to quantify the magnitude of gene expression changes between treatment and cultural conditions, and show various gene expression levels over treatment period. Positive value indicate hunger unregulation, mean gene expression in the treatment graph is higher than the in the control group. Negative value indicate then regulation, mean gene expression in the treatment group is lower than in the control group. This polyphenol allows for a quick assessment of how target gene expression is affected by treatment. Log FC is typically used in combination with statistical significance testing, such as the p-value or just the p-value. P-value indicates whether that change is statistically significant. A gene is generally considered significant differentially expressed when the absolute value of log Ft is greater than a certain threshold, and the p-value is less than 0.05. In this case, the mRNA expression of Ki67, PCNA, and PGP genes is not statistically significant under those treatment conditions for the dataset. I will further prove the mRNA expression of those genes in my experimental setup using the co-constraint-rich subtraction of random pure ethanolic crude extract compared to the pure costing in the selected cell lines. My expected outcomes. This project is expected to select one or a few cancer cell line rather than those usually used in previous study, such as prostate, cervical, lung, breast, and colon cancers. The experiment will also prove the costing-rich subtraction of random pure our crude ethanolic extract from random pure will have a stronger inhibitory effects on the growth and the proliferation of selected cell lines compared to pure costing. Subsequently, both compounds will prove the reduction of fusion expression associated with cell proliferation, and drug resistance within 2, 5, 12, and 24 hours. My further work is to prove the growth inhibitory effects of the costing-rich subtraction of crude ethanolic extract from the random pure compared to pure costing on the selected cell lines HEP2 and K562 using the MTT or LDTSA. Subsequently, the mRNA expression of Ki67, PCN, and PGP genes associated with growth inhibitory effect and drug resistance on the selected cell lines will be evaluated using PCR. This is my Gantt chart. it's my reference, and thank you for listening.
All right, thank you, Ziyu. So now we can open up for questions. Dr. Chu and Dr. Shahida. Dr. Chu, you're welcome to go first if you like, following the order.
Okay, thank you, Dr. Kumita. Sorry, Zheyu, I thought this is the final presentation, right?
Yeah.
Uh, okay. Oh, so okay. So you have to present again later, or?
Yeah.
Oh, okay.
I-
So, my first question will be, in your third slide, right, you mentioned like you mentioned something like well documented but not well studied. The slide number 3. What do you mean by that?
Oh, it means that most of the analysis will has been studied by researchers, but a few analysis was not have the very well studied. There are more than information we can get from the the a little sunlight.
Okay. Then my next question would be, your expected outcome is you expect that your compound will be perform better than the pure quercetin, right? You put the expected outcome. What do you base on? What is the basis for that statement? Why do you expect that your compound will perform better than the pure quercetin? Based on what reason?
Could you repeat?
Yeah, because you put your expected outcome, you expect your compound to perform better than the pure quercetin, right?
This
The second one.
second one.
No. The correct. The slide is, go back to the slide. The expected outcome slide.
Oh.
Yeah, the second statement.
Oh.
You expect that your red onion peel, the quercetin extract from your red onion peel, will have a stronger effect than compared to the pure quercetin. Why do you think so?
According to the previous article, the previous article proved cosine can can it have the inhibitory effects on the sunlight and have yeah the most of the article I saw said this it can it can inhibit the cancer sunlight. So I want to prove that again.
Okay. Then I think my final question would be, may you share with us what is the major obstacle or problem that you face in conducting this research work? What kind of problem you face, and what kind of effort that you can do to complete the project?
Oh, please repeat.
So far,
Oh,
seems like you have faced some obstacle or some problem, right? I mean, that delay your work. So what kind of problems that you have faced so far? And what can you do to overcome those issues?
uh, actually, I don't know. My understanding for this question is clearly, and I often use the GPT and search article to solve their issues. And of course, my professor also helped me. May I understand wrong?
Yeah, because we are just want to make sure that you finish the project on time, right? So you just want to know a bit, so you can share with us your maybe your problem or whatever that you face. That then, what can you do to make sure that you can complete, if you can finish the project on time? That just some
I
sharing,
think
so that we want to know what kind of effort that you can put in to make sure that you complete the project on time.
I think not not on time this semester. I must extend the other, and I will try my best.
Okay, Dr. Komita, I'm done with my questions.
All
Thank
right,
you.
thank you. Dr. Shahida, do you have any questions?
Uh,
All right, thank you, Dr. Kumi. Hi, Gao.
I
Yeah, can you show us your slide on objective? Okay, identify, analyze the expression, and so far, you have completed, I guess, the objective number 1, right? Hello.
hello.
Yeah.
This one.
From this objective, which one have you completed so far?
These two, 1 and 2.
This 1 and 2. I mean, oh yeah, okay, the geo. And you have left with one more objective, which is to run real-time PCR and so on. So from these two objectives, what can you conclude from your findings? I mean, the conclusion that which cell line, can you just repeat the question that you have presented? Hello, hi Gao. Do you hear me?
Yeah.
Okay.
So please repeat. I also
Okay, just now you mentioned you have completed 2 objectives,
Yeah,
the first and the second one.
yeah.
So from your analysis, what is your conclusion from the analysis that you have done so far, the geo and the first one? So what is your conclusion?
Mm, from the executive aspect, uh, the first analysis can be used to my study, but I think the only one is the most sensitive one. So I used I only used this online, and uh, and but the geo results actually is not very well. You can see the this violin map is similarly, and the HapG and the HapG2 satellite from the GEO is also is not very different differentially. Um, but I believe Cousin can inhibit the satellite. Just if we found the data is not very real. Yeah.
Okay. And then do you put your methodology for real time and gene expression study? Yeah.
Which what?
Can you go to your methodology? Okay, with
This
this
is
one.
methodology.
So this one you haven't performed, right? You are going to do after this. Have you
I have
done
gone
this
to
one?
what?
Have you done this one?
Oh, you mean the cell proliferation? I will do this work in.
So in this means that you will have another final presentation to discuss all your results, right? Because you haven't performed. I think there's a lot more that you haven't done, and do you think that you can complete all the experiment within time?
So I extend my semester and faster to them. This week I will do the LDHC to anti-proliferation. Yeah.
Yeah. Yeah, I think I look forward to your next presentation to see all your results then. I think that's all from me, Dr. Kumita.
All right, thank you so much, everyone. I think that marks the end of the session. So we will end here right now, if that's okay with everyone. Thank you so much again, everyone, for being at the session. Have a good day, everyone.
Thank you.
Thank you.
Thank you. See you.
All right,
Bye.
thank you. Bye. Puan Ari, kita boleh end dah kan?
Boleh doktor.
Alright, thank you so much. Bye,
Okay, doktor.
everyone.
Thank you. Bye. Dah habis dah. Siapa yang di stop? Habis stop. Ni. Dah dah. Habis dah. Stop.